Hi everyone. So we have lab 4 here. Introduction. Organelles are membrane enclosed vesicles inside all eukaryotic cells that function in a variety of important cellular processes. We shall isolate various subcellular fractions from mammalian liver by a technique termed differential centrifugation. We will then assay these fractions for succinate dehydrogenase, a specific marker enzyme for mitochondria. So the fact that succinate dehydrogenase is a marker enzyme for mitochondria means that it should only be found in the mitochondria. This laboratory is an introduction to methods of cell fractionation using differential centrifugation and enzyme assays for analysis of cells and tissues in molecular biology. So this information should definitely be in your introduction. Okay, so when you tell me the purpose of this lab was to do blah blah blah, and then the techniques that we used to accomplish our goal were differential centrifugation and enzyme assays. The arrangement of macromolecules within a cell is as important to cellular function as their catalytic activities. Cellular compartmentalization provides efficiency by bringing together related compounds, like the enzymes of the mitochondrial electron transport chain, or by separating those compounds that can interfere with each other, i.e., lysosomal hydrolytic enzymes. So, for example, inside of a cell, we need the organelles to be separate because they each perform their own independent functions. For example, lysosomal hydrolytic enzymes have things that will, if those get out, then they're going to start digesting the rest of the cell, and then your cell's going to die. So that's why it's important to compartmentalize. Cellular compartmentalization is, in part, accomplished by various subcellular organelles. In this lab, we will isolate several subcellular organelle fractions from liver cells, and we'll examine their various properties. The method we will use to separate the various organelles utilizes differential centrifugation to isolate components of different densities. So, if I were to ask you, how does this technique of differential centrifugation work? Well, it isolates components depending on their density. Okay? With this technique, the heaviest or most dense organelles will pellet in less time under lower gravitational forces. So for example, we have our centrifuge tube here, and it says that the nucleus is really heavy. So our first centrifugation was 1000 RPM for 10 minutes. And so we got a pellet down here, and then we had supernatant. The supernatant is the liquid. So they're saying that after our first centrifugation of 10 minutes at 1000 RPM, we were able to pellet the nucleus because the nucleus is more dense, okay? But the mitochondria was still hanging out in the supernatant, okay? So with this technique, the heaviest or most dense organelles pellet in less time under lower gravitational forces than is required to pellet lighter organelles, such as mitochondria. So in order to get the mitochondria, because they're lighter and they're less dense, in order to get them to centrifuge, we're going to need to increase the RPM and the amount of time that we centrifuge, okay? So first, a cell homogenate is made by breaking open the cell membrane. The homogenate is then centrifuged for a short period of time to pellet the heavier components. So remember that we started off with a little piece of liver, and then I went ahead and chopped it up for you, and then I gave it to you, and you went ahead and put it in a homogenizer, and then you squeezed it with a pestle to break apart. We had cells here, so you went ahead and broke apart the membrane in this manner. Okay? For this type of fractionation experiment, which tissue we use and the method of homogenization are dictated by the biological system. Homogeneous cell populations from cell culture are well suited for cell fractionation. Some organs, like the liver, also have one cell type that predominates, so they are also well suited. Once a cell type is chosen, liver in this experiment, it is important to obtain the organelles in a biochemically active, morphologically whole state. So basically what they're saying is that we have a cell and we want to break it open, but when it comes to the organelles like the nucleus and the mitochondria, we don't want those to be broken. They have to be biochemically active and morphologically whole. That means that they need to be perfect so that we can actually test them for what we need to test them. If they're broken, then our experiments may not work. So the homogenization buffer often includes sucrose to partially dehydrate the organelles, keeping them intact. No technique used to isolate organelles is perfect. It is very difficult to get pure, unbroken preparations of any organelle. Techniques providing optimal isolation of one organelle may completely rupture another organelle. Thus, methods are often used to measure the contamination of one organelle fraction with another. Analyzing each organelle fraction for organelle-specific marker enzymes can accomplish this. So that's why we're using a succinate dehydrogenase, which is a marker enzyme for the mitochondria. Organelle assays. We will evaluate the purity of our subcellular fractions by analyzing each fraction for succinate dehydrogenase, the marker enzyme for mitochondria. It is scientifically well documented that some enzymes are located specifically within certain cell fractions. Okay, so here's a table for cell organelles, their size, and then their density. So I highlighted the mitochondria. The size is about 1 to 2 microns, and the density is 1.1 grams per centimeter cubed. Over here we have another table, and it's organelle density and unique markers. So for the mitochondria is succinate dehydrogenase, and that's the marker. Then we have, when interpreting our assay results, it is important to remember that the activity of each organelle fraction can be influenced by a variety of factors like diet, age, sex, and extent of fasting of the host animal, and also by the experimental method used to obtain the fraction. It is also important to note whether a specific enzyme is found in significant quantities in any cellular fraction that should not contain it. So for example, the succinate dehydrogenase is a marker enzyme for mitochondria. So if we find it anywhere else, then it means that something went wrong because that enzyme is not supposed to be anywhere else. So for example, if your cytoplasmic fraction is found to contain large quantities of the mitochondrial enzyme succinate dehydrogenase, then contamination of the cytoplasmic fraction with mitochondria, or lysis of the mitochondria and release of their enzymes into the cytoplasmic fraction has probably occurred. So that means when we were using this pestle to break up the liver, it's possible that maybe we were too rough and maybe we broke the mitochondria as well. And that's why we have contamination. It went everywhere into all the fractions. So quickly, I'm going to go ahead and mention the fractions that we have. So we've got a homogenate fraction, the nuclear fraction, the mitochondrial fraction, the post-mitochondrial fraction, mitochondrial B, and microsomal. Okay. Succinate dehydrogenase assay. Mitochondria are the eukaryotic organelles that contain the enzymes of the citric acid cycle, the electron transport chain, and oxidative phosphorylation. The organelles composed of an inner and outer membrane. the outer membrane is relatively permeable, allowing most molecules with molecular weights up to 5,000 daltons to pass into the inner membrane space. The inner membrane, however, is very impermeable and allows only small uncharged molecules to penetrate into the matrix. The enzymes of the electron transport chain and oxidative phosphorylation are embedded in the inner side, the matrix side, of the inner membrane, with the exception of succinate dehydrogenase, which is located within the inner membrane. All the enzymes of the citric acid cycle are located within the matrix. Mitochondria can be prepared from a variety of eukaryotic tissues, both plant and animal, by differential centrifugation. They are pelleted immediately after removal of the nuclei. The presence of mitochondria in an isolated subcellular fraction can be verified by using mitochondrial-specific marker enzymes. In these assays, care must be taken to choose low molecular weight, non-ionic substrates and dyes, which are capable of penetrating both the outer and inner mitochondrial membranes. One mitochondrial specific property that is easily assayed is the ability of the enzyme succinate dehydrogenase to reduce the dye DCPIP, dichlorophenylindophenyl. Succinate is used as the substrate, and its oxidation to fumarate requires an active succinate dehydrogenase, which is located within the inner membrane. Normally, in the absence of exogenous dyes, the reaction occurs with a concurrent reduction of the coenzyme FAD. Okay, so here we have to remember what is reduction and what is oxidation. So reduction is when something gains electrons, and remember, electrons are negative. And then oxidation is when something loses electrons and becomes more positive. So with these types of reactions, the hydrogens are the ones that carry the electrons. So if we start with succinate, we need to know if this is the reduced or the oxidized version. And then fumarate, we also need to find out if it's the reduced or the oxidized version. So if we look at H for succinate, HOOC, over here on fumarate we have HOOC, nothing's changed. But then over here on succinate we have CH2, so there are two hydrogens, which means that there are two electrons. And then over here for fumarate we just have CH, so there's only one hydrogen, one electron. So because succinate has two electrons, it is more negative, right? So this is the reduced state of this molecule. Succinate is the reduced state. And then fumarate has less electrons, which means that it is more positive, so it is oxidized. Now, we look at FAD, and over here on the left-hand side, FAD does not have any hydrogens, so it doesn't have any electrons. But over here for FAD on the right, it has two hydrogens, so it's carrying two electrons, which means that it is reduced. And on the left-hand side, it is the oxidized version. So succinate dehydrogenase is the enzyme that is responsible for doing that. So instead of using FAD for our experiments, this is where our dye is going to come in. And we're going to go ahead and use DCPIP instead of the FAD. So on the left, we have the DCPIP with no hydrogens, no electrons. On the right, we have the DCPIP with two hydrogens and two electrons. So this would be the reduced state. So succinate dehydrogenase makes the reduction of DCPIP H2 possible. So let's read about that. However, during an assay for succinate dehydrogenase, an excess concentration of a dye, usually DCPIP, is used to observe its reaction. Reduction of DCPIP during the reaction decreases its absorbance at 600 nanometers. It turns from blue to colorless. The concentration of substrate, succinate, is also used in excess to prevent the reverse reaction. The reduction of fumarate with simultaneous oxidation of the dye. So in the present experiment, we will use the succinate DCPIP oxidation reduction assay to test various liver subcellular fractions, and those are the 6 fractions that we talked about earlier, for the presence of active succinate dehydrogenase. Okay, so now that we know what's happening and what we're testing, so basically if we have a lot of DCPIP in a sample, and remember we have 6 fractions, so we're going to test each of them for the presence of this enzyme, succinate dehydrogenase. But if one, for example, if one fraction has DCPIP and it's blue, but it is never reduced, so it never gains the 2 hydrogens or 2 electrons, then that means that succinate dehydrogenase enzyme is not present, which it shouldn't be. In most of the samples, it should not be present. It should only be present in the mitochondrial fraction, right? Because succinate dehydrogenase enzyme is specific. It's a marker for the mitochondria. So that could be a really nice hypothesis. Like I hypothesize that out of the 6 fractions, we will find the presence of succinate dehydrogenase in the mitochondrial fraction. And then you have to tell me why. Why do you think that? And then so what you can do is describe this reaction. That would be a really nice scientific hypothesis. Section A, liver fractionation. Introduction. The liver is a favorite organ to employ to investigate many metabolic enzymes. The rationale for using liver tissue is that its cells are highly specialized for metabolic processes, and the abundance of these components makes it straightforward to obtain enough working material from small samples. So you can put that in your introduction, talk about the liver and how a lot of the cells are really similar in the liver, and it's really easy to like break it up and get a lot of the enzyme that we're looking for. The protocol must be conducted at ice-cold temperatures to minimize the activity of detrimental components of cells, which are freed during the fractionation process. One reason for possible ambiguous results from this protocol is failure to maintain a low temperature. Keep all samples ice cold as much as possible. Activity A1, tissue homogenization. So each group of 4 shall be provided with a piece of mammalian liver. You're going to weigh it, make sure it's 1 gram, and then you use a homogenizer to break it up. So that's what we did. Activity A2, then we did a nuclei isolation. This was our first centrifugation at 1000 RPM for 10 minutes. And we had the pellet that was the nucleus, and the mitochondria were still in the supernatant. And then activity A3 was the mitochondrial isolation. So because the mitochondria are smaller, we went ahead and increased our RPM to 10,000 for 20 minutes. And this went ahead and pelleted our mitochondria down here. And then we ended up with all of our fractions. We had 6 fractions. We stored them at -20. So these are our fractions. We had homogenate, nuclear, post-mitochondrial, mitochondrial, mitochondrial B, and microsome. So week 2, introduction. Enzyme assay. Last lab, you isolated various cellular fractions from liver cells. Each of these fractions will be assayed, this time to determine the degree of succinate dehydrogenase activity. In In reality, just one of the 6 fractions should possess such activity. Should the other fractions show any activity of this enzyme, that will indicate to you that there was some disruption and leakage of mitochondrial intermembrane materials. Okay, so that's your hypothesis is, you should find it in one of the fractions. And then in your discussion, so remember, in your results, you're only going to tell me facts, exactly what happened, and that's it. Keep the facts. In your discussion, that's where you're going to explain things to me. Okay, so I found this in the nuclear. Succinate dehydrogenase was not supposed to be in the nuclear. I had contamination, and then tell me what could have happened. Like maybe when you were breaking the liver cells apart, maybe you were too rough and things broke and the mitochondria ended up in all the fractions. Like that could have happened. Like tell me what else went wrong. Activity B1, succinate dehydrogenase assay. So this is what we did today. So turn your spectrophotometer on, 600 nanometers. Recover your 6 tissue fractions, and we set up our little cuvettes like this. Use cuvette G as a blank. We did that. We worked quickly. Okay, and then that was it. Analysis. Okay, so for your analysis, you have to plot enzyme absorbance at 600 nanometers versus time. Combine the data for all 6 cellular fractions on one graph. Okay, so that's going to look like this. Let me draw it out. Okay, so your thing that you're going to turn in should look like this. On the y-axis, we have absorbance, wavelength. So you're going to put at 600 nanometers instead of wavelength, okay? Absorbance, 600 nanometers. And then on the x-axis, you're going to have your time in seconds. And you're going to go ahead and do all 6 fractions, so all 6 lines on the same graph. So it'll probably look something like this, and make sure your lines are labeled. This is going to be submitted for your results, so you got to do this on Excel.
Hello everyone, sorry I'm working from home, so you're going to hear some background noise, but it is what it is. Here I want to show you how to set up your Excel sheet. So we have our homogenate, nuclear, post-mitochondrial, mitochondrial B, and microsome fractions. And then, let's see, I was practicing up here. Okay, and then we have our time intervals. So this was 5 seconds, and then 10 seconds, and then 15, and then 20. And then what you can do is you highlight this, and then you drag all the way until the rest of the time points that you need. It was 4 minutes times 5 seconds. So however many that is. Okay. So then you go ahead and fill in your values. So if it was like, if you started at 2.345, 2.300, 2.2. Okay, so I filled out 2 of these lines, and then some more information up above. We have 4 minutes total, and so 4 times 60 is 240 seconds, divided by 5 is 48 time points per sample, because we documented the readings every 5 seconds. So then the way to, you have to keep going, of course, because you have more time points. I'm just giving you an example of what to do. So then you grab and you highlight whatever you have. Okay. And then you click this little guy right here. and you say charts, and then you say line. Okay, so we've got our graph. And then we go to the plus icon. And we're going to remove the grid lines. And we're going to go ahead and add chart titles. Oops, chart title, axis titles. There we go. So this is going to be absorbance. And it's going to be in microns. So we're going to go to insert, symbols, symbol, micro, insert, close. So micrometers. What's microns? Oh no, it was in nanometers. My bad. Sorry. Absorbance in nanometers. It was 600. 600 nanometers. Okay. Alright, chart title is, so this is absorbance over time. Okay, we can close this. And then this axis title is time in seconds. Okay, and then you see how it has its own, like in blue we have the homogeneate, and then in orange we have the nuclear. So once you fill out all your numbers, whatever you highlighted, that's how many lines will be here. So for you, I think it'll be 6 lines, and it'll just do that part for you. And then what you do is, we need more information. So we go to the line, and then I think you right click, and you can say add trend line, and then display R squared value on chart and display equation. Okay, so we're going to do that for each line. So there we go. For this orange line, we have that. Okay, and then this is our slope right here. This 0.0712x. So this negative 0.0712. That's the slope for this line. So again, I'm going to do it in the blue now, just as a second example. You click on your line, and then you right click on your line, and then you say add trend line. Okay, and this window just pops open, and we're down at the bottom, we're going to click display R squared value on chart and display equation on chart. Then we're going to exit out of there, and then here we go. So let's see if we can make this bigger on the right. Let's see if we can make this. Yeah, okay, so you can play with the lengths of these so that there's more space for your formulas to show up on the right. So you just click on this portion and you bring this in a little bit, and then we can click on our equations so that they actually show up. Okay. Okay, so that's the slope for this one, that's the slope for this one. Okay, when you look at your Lab 4 protocol, there's this section called Analysis, and this is what you have to turn in for your results section. So plot enzyme absorbance at 600 nanometers versus time. Combine the data for all 6 cellular fractions on one graph. So I just showed you how to do that. And then calculate the absorbance per minute. So this is the slope of the graph. So you have to do that for each fraction, and I showed you what your slope was. And then should the slope not be linear, perform your analysis at the start of the reaction where the slope should be more uniform. But if your line is pretty linear, then we can take that as the slope. You don't need to go any further. But if it's really not linear, then you can go ahead and just take the beginning of it, or whichever portion is more linear. And then C, you have to compare the activity per mil of extract and show your calculations. So for this, there's a document on CAT courses. Okay, so I'm in your Lab 4 documents. That's the cell organelles PDF, pre-lab. It's this document right here, calculations, advice. And it says you will need to turn in a graph and enzyme activity calculations with this lab. The graph will show the absorbance at 600 nanometers on the y-axis and the time in seconds on the x-axis. Make sure to give your graph a title and a legend. Which line is equal to which fraction? So the whole legend thing, Excel did that for us. You see down here, the homogeneous is blue, the nuclear is in orange, so Excel does that for you. To compare, let's see, you will also need to include a best fit line for each fraction to obtain a slope. So we did that. To compare the activity per mil of each extract, you must graph your data as stated in the protocol and a best fit line drawn. So we did all that. The slope of this line represents the activity of the enzyme. Okay, so our slope is, for this orange line, is negative 0.0712. So that is the activity, that slope represents the activity of our enzyme. Okay, and for the blue line, it's negative 0.0732. Okay. To compare the activity per mil of the extract, we must consider the mil of each fraction that was added. Note, different volumes were used for each fraction. The total volume for each cuvette was 3 mils. So to calculate the enzyme activity, you will divide your total volume by the volume of the fraction added, where your enzyme should be present. Then you will multiply that value by the absolute value of your slope for each fraction. And then they give you an example. Cuvette A, we had 3 mils of total volume divided by 0.5 mils of cellular fraction homogenate. So for your calculations, this remains the same because we had 3 mils of total volume in all of our cuvettes. And we also added 0.5 mils of cellular fraction for all of our samples. So we're going to do 3 divided by 0.5, and that's going to equal 6. And then we're just going to multiply 6 times, oh sorry, that's my dog. We're going to multiply 6 times our slope. And because we have these lines right here, that means that our slope is going to be only a positive number. So 6 times whatever our slope was, and that's going to be the answer. That's going to equal the activity per mil of extract. Okay, and you're going to do these calculations for all 6 lines. And you can add them down here or wherever you want. So in your results section of your lab report, you're going to add your graph, and then you're going to tell me all of these values. So this is probably going to be a longer results section, 2 paragraphs. So your first paragraph, you can go ahead and describe what we're seeing here. You can say, you know, figure 1 shows absorbance over time at 600 nanometers. And then say values, absorbance values were recorded at 5-second intervals, you know, but like in your own words. And then say, remember, it's only facts. Say, you know, fraction 1 was, or the homogeneous fraction had a slope of blah blah blah, you know. Fraction 2, or the nuclear fraction, had a slope of blah blah blah. And then you can say it had an enzyme activity per mil, you know. You can answer the questions that were asked here in A, B, and C. Compare the activity per mil of extract. So you can say, you know, I guess you can say the homogeneous fraction had a slope of this, and it had this activity per mil. The nuclear fraction had this slope, and it had this activity per mil. And then you continue that way for the results. And then in the discussion, that's where you talk about, what does this mean? Was your hypothesis correct? And you know, what were your expected results? We expected, and then also, sorry. Okay, so also you may want to include a picture of your results. So this was the homogeneous fraction, this was the nuclear, this was the mitochondrial, and you know, what was your hypothesis? Succinate dehydrogenase is a marker enzyme for the mitochondria. So we would expect, if the experiment went perfectly, we would expect that we would see a lot of succinate dehydrogenase activity in the mitochondrial fraction, because that enzyme is a marker enzyme for the mitochondria. So it shouldn't exist anywhere else. And then if the way this assay works is, you know, if we have succinate dehydrogenase activity in one of these fractions, then we would expect the color to go from blue to clear. Okay, and if it goes from blue to clear, that means that you have succinate dehydrogenase activity. So if you hypothesized that you would find most of it in the mitochondria, or the mitochondrial fraction, then our hypothesis would be incorrect here. So the one that went really clear was a nuclear fraction. So we're seeing the most succinate dehydrogenase activity in the nuclear fraction. So for your discussion, this is where you talk about what happened for each of these fractions. What did you observe, and what does it mean? So here in the homogeneous fraction, we wouldn't expect any succinate dehydrogenase activity, but it's light blue. There's actually some activity. In the nuclear fraction, we have a lot, like a lot. And then in the mitochondrial fraction, we don't have anything. And then in the, I forgot which fraction this is, it might be post-mitochondrial, but there it's light blue, so there is activity here. So for each of these in the discussion, you can kind of like group them together, or like these two can be grouped together. You can say in this and this fraction, there's almost no activity because the color is pretty dark blue. And you can say what that means, you know. Okay, I think I walked you guys through everything. So if you have any more questions, please come to office hours. There is no lab next week because we have a Thursday holiday. So the lab reports are not going to be due next week. They will be due the following week. And yeah, if you need help, come to office hours.